Hey everyone, Assalamualaikum and welcome to the Maths, where education meets excellence. And I hope all of you are doing fine. Please give strict following to the SOPs because of the spread of coronavirus. Let's go back to your topic, and that is repair by homologous recombination. This is also a mechanism present in our genome that helps in the repair of DNA damage. So how does it work? So there are many genes, okay, which are responsible for your homologous repair. Okay, number one of them is your ATM. Then is your BRCA1 and BRCA2. Okay, BRCA, I call it BRCA1 and BRCA2 to make it easier. So what happens in in this type of cancer, okay, in which a lot of mutations accumulate. So for example, there is a lot of radiation, okay, lot of radiation. We call it ionizing radiation, and this is going to mutate your ataxia telangiectasia protein kinase. That is going to result in your ataxia telangiectasia, which is a neural disease. And then if there is some alkylating agent, okay, alkylating agents are actually very reactive, okay, and these react with virtually everything inside your body they can react with. But what do they do in your DNA? These are actually also very smartly used in a controlled way to cure cancer as well. Okay, so enough about that. What do your alkylating agents do to your DNA? They introduce double-stranded breaks, okay, and these breaks are repaired by your p53 related mechanism. But if there is some alkylating agents, it's going to make your DNA unstable. There will be double-stranded breaks, and this is actually going to cause a syndrome called as Fanconi syndrome or Fanconi anemia. Okay, then it's also going to cause another syndrome, and that is called as Bloom syndrome. So like its name, it's not going to bloom something, obviously. It causes developmental abnormalities, and I think perhaps that is why they called it Bloom syndrome. And in Fanconi there is anemia. So these are some things which are related to your homologous repair, which need homologous recombination, ionizing radiation and alkylating agents. Okay, these agents cause formation of dimers. Okay, these cause from the dimers are formed by a mechanism which is cross-linking. So it causes dimers, it causes breaks by cross-linking your entire DNA sequence wherever they want. Okay, they are not specific. If they were specific, we could make medication which were specifically directed at them. So what happens actually, this is your DNA, okay, and here is a cross-linking mechanism happening here. So if I magnify it for you guys, what happens here, our body recognizes it. For example, ATM protein senses it, signals your p53 protein, and this p53 protein tries to, what does p53 do? It tries to repair it, and repairs it how? So it's going to look in your entire genome. So this is your entire genome, okay, and it's going to search here, it's going to circulate and search here, and will find, okay, not p53, your body is going to do it on your own. It's going to find a matching sequence here, and it's going to bring this sequence close to this broken strand. And if it's correct, it's going to attach the strand in your DNA. Okay, so this strand, which was actually very similar, this strand is called as homologous strand. Okay, homologous sequence. So now you know why it's called homologous recombination. So if, for example, your maternal strand is damaged, your body is going to look for a good strand, a similar strand, a homologous strand in your paternal genome, which you have inside your body, and going to bring it closer to the damaged part and then going to attach it. Okay, so this is your homologous repair. Okay, and this is how it normally works. But there are mutations, obviously, and these mutations are usually related to ATM, that is your ataxia telangiectasia protein kinase. Okay, and an interesting thing is that BRCA1 gene also is involved in this. So if this protein is mutated, okay, there would be ataxia telangiectasia. What about BRCA2? BRCA2 gets associated with a gene which is RAD51, and it's going to suppress Fanconi anemia. Okay, normally, but when both of these get mutated, there will be Fanconi anemia. So I associated BRCA1, BRCA2 to your DNA repair system, even though they are tumor suppressor genes. Okay, but they play a part in your DNA repair as well, and this is how they do that. They fuse with your ATM, they fuse with your RAD51. Okay, so this is their part. Also, BRCA1 and BRCA2 are the leading causes of breast cancer in female. Okay, and as you know, it's familial, runs in families, and 50% of the cases of the breast cancer are familial, and they have mutation of BRCA1 and BRCA2. Also, they can cause prostate cancer in males. Okay, prostate cancer, and the involving protein is BRCA1. Then also, the BRCA2 can cause a lot of cancer. For that, I have a mnemonic for you guys, and that is B as pops. Okay, B as pops. Pops is a thing which you eat. Okay, pops. I loved them in my childhood. So for B, there is breast cancer, obviously, but this B here stands for bile duct cancer. Okay, bile duct cancer. Then S is for stomach cancer. Then P is for pancreatic cancer. O is for cancer of your ovaries, and P is for your prostate cancer. Also, it causes lymphocyte cancer as well. Okay, so this is your homologous repair, homologous recombination repair, and the diseases associated when the genes in the homologous repair are malfunctioned, which I told you: ATM, BRCA1, BRCA2. Okay, so this is it about today's lecture, and I hope you understand it better now. And if you don't, you can ask me questions right now in the comment section. And if you like this video, give this video a thumbs up and subscribe to our channel. Share this video as well, please. And I will see you in the next one.